Hello and welcome back to another episode of Pyblock. I have made a big decision here. It is to skip the whole kill calc growing thing because it's going to require a ton of things. So this is the killer here. Biosample the cDNA. This stuff is just crazy complicated, and this is what's some of the stuff that's going to be needed for green science. So I might as well hold off. Just do the fiber recipe from wood, and we're going to do a big wood farm. So I've started off with some CO2 production, get some moss going so we can plant our tree seedlings. Let's see, let's get this going because I need to some other tree seedlings going here. Let's see, let's put the seedlings in there. Botanical nursery. This is gonna feed our So you tree seedlings. Put a bunch of those in there. Water. I need water up here. Let's pull it off of this pool here. Water. And our moss. We're gonna feed this moss up here. Moss. Just gonna get those going. Let's do a quick and dirty. Extra trees production there. Rest of those can go down here. Let's see this stuff. Water on the outside. Some extra water production here. Water, there we go. Add some power for these things. Okay, that's water. Now we need our input there, and our output there, and output there. Do a big sawing system here. Okay. Your wood recipe. Okay, so these guys, your trees, growing faster. There. So actually a reasonable speed. Yeah, this is working out perfectly fine for carbon dioxide for these guys. All backed up. Like I need more tree seeds at some point, but get this going. Let's run this up here. this wood output. These guys can be close side of the belt. Go. And do that guy, a tree seed. Grab from there. Okay. And we get our wood output. Put it here. And that's wood production. And we can expand it way out if need be, which we probably will need to. Okay, so this will get split. Some of it's going to go down here. To this wood production, the treated wood production. Other portion going to come up here, make it into fibers. There's this fiber recipe. Fiber. Send the circuits, yes it is. Wood processing unit. More of those things. Just to start off with, redoing this recipe. There. Fiber. Okay, that'll take a lot to do there. Tap into the steam from over here. Steam pipe is this one. Steam. Okay, we're making our boards. I'm gonna need an output here. Let's do this. There we go. That's board. We just need to mix it with that melamine resin, which is a whole nother thing. Yeah. Yeah, urea, formaldehyde. Till we can get animals going, we'll have to manually manufacture those things. Right. Can I do moondrop stuff? Do I need this? Drop from moondrop seeds. Yeah. Okay, what's the other methane making recipe here? Just the moondrop stuff. Gas bladder, manure. Yeah, I'm not at coal processing 3. Or am I at oil processing? Nope. Not that. Really? I have to get into that stuff to get circuits going? Oh boy. I was hoping not to. But Only place to get the resin. This stuff, formaldehyde. Oh boy. And we need that melamine and urea. Some other methane making thing. Formaldehyde. No. I have to get into the cDNA stuff. I got some boards made anyways. Yeah, phytoplankton. It's like some other stuff here, manure. Wait. I need that Zognobacteria. Oh, that's just that. Make that stuff. Got moss, seaweed, biocontainers. Wastewater comes from the phytoplankton, I think. That process. Funbe. Phytoplankton farm. Yeah. Retrovirus, plasmids. You can have all that stuff. You don't even have the retrovirus thing. Ah, there, genetics. Gene lab. Retrovirus. Wait, what? Were you able to get Verox without these things? Hold on. Oh yeah, no, hold on. Verox stage 1. Okay, yeah. Do have to get the Verox thing. Ah, man, stuff is complicated. Just to get circuits going. Where are these Verox not? Some science in there, please. Finish that. Let's get the genetics going. Okay, Verox stage 1. It's AOGs. Some glassware here. Okay, Frog, plus a cage, yeah. Paddock. Paddock. I need plastic. Where do I get plastic? Let's see here. Got like oil stuff. Plastic. Plastic bar. Syngas and aromatics. Biofactory. Okay, let's make that. Since this is going to be down this rabbit hole. Biofactory, where's that? Biofactory, Nexalite. Looks like I need to use some of this Nexalite we've been making here. I have to wash it. Nexalite. Wash a bunch of that. Slap a sinkhole on the front. Oh, I'm out of parts. Sinkhole. Clean Nexalite. Now what do I do with that? Just furnace it. Okay. It's 3-to-1. That makes 6. This makes 18 plates. Yes, okay. Foundry, we've got one of those. The next light plates, please. Go. Okay, biofactory. Put this plastic on the bus, I think. Be fine. So we got tar right there. We can tar processing unit. Aromatics. Grab some syngas. Our syngas production. Closer read to that. Grabbing that stuff. Syngas is over here. Here it is. Already. Oh yeah, I have it going up here. Quite know where I'm routing this. Up here somewhere. To the side, maybe. We'll do that. Okay. We've got tar here. Let's do our tar processing unit. We make the creosote. Make the aromatics out of that. That stuff. Then we get this biofactory. Plastic bar. Let's reorientate that. Plastic bar. Power. Syngas goes in. We get plastic. Okay, don't have anything on these lines yet. Okay, plastic. Now that we got the plastic, we need to make maybe, what is it here? Some more steel. You're running out of aromatics. That's being a bit slow. It's okay. Steel. Varrock paddock. Okay, we can make a. See, the Varrock is going to require some biosamples, right? Where is it here? Orcad, no. Here we go. Generic Earth DNA samples. Where are those? Those ones, just biosamples, science. Why are you not operating? Okay, did not put those in. Okay, should get some more science now though. In a little bit. Bunch of containers there, like some of those too. Cages. Spoons there. Sap, biomass, moss. Not too bad. That codex. Just need a bit of glass. Yep. Codex, moss gene samples, biosamples. Yeah, I need to get those things going. Let's build an incubator. Incubator, right, that's another thing I need plastic for. Bunch more glass. Okay. Grab this plastic here. Actually, this should just be full of plastic. Okay, let's make that incubator. Connect the light. All the resources. There. Incubator. This. Put some wood in there. Get an air pump. Have one already. Pressurized air. And then I just need to make some lamps. Okay, bacteria. Combine it with okay, this stuff does make my brain hurt a little bit. So for these biosamples, so the phytoplankton, we have our phytoplankton farm. Iron ore and water. Let's see, iron ore. Route some ore this way. Okay. Divert some of that ore we're making. Make some phytoplankton. And we're almost there on this stuff. Seaweed and moss. We can divert moss and seaweed from here. Moss. Moss and seaweed. Biosample. This needs to be made on a biofactory. Okay. Biofactory. Feed those things in. Sogna. Phytoplankton. Wastewater. Wastewater comes from fish. Ah, yes. Fish farm. Some seaweed and saline water. Got seaweed here. Tap into the seaweed water. The saline water. Seaweed water. See, I was tapping into it over here, right? Yeah. Tap into this. Saline water will make a fish farm. Just the fish. We only have the basic fish farm. Yes, we do only have the main basic fish. Oh, okay. Saline water, yeah, that does reduce the time decently there. Okay, fish farm. Might as well do saline water. Need to catch some fish real quick here. Fish. Go. Give it some saline water. He starts making fish. Okay, and we get our wastewater. Again, this is not going to produce very much, but it's okay. It'll just get things going. Something down, you know. Fish into a chest there. Fish, fish, fish. See, extra fish. There we go. That made wastewater. Now I just toss in my bio containers and we get bio samples. Okay. There we go. Bio samples. Now what else do we need for this guy here? So the generic Earth DNA samples. So those bio samples and science. Let's grab science here. Some of them researching there. Generic DNA samples, which has to be made in the fancy machine over here. Generic Earth DNA samples. Good. More of these containers here. More glass. Need so much glass here. This stuff. A few of them. So, this guy, creature chamber, petri dishes, some more biosamples, and moss gene sample. Alright, gene lab. Need to finish up genetics there. That. Drink some water. Oh, and we are over the 30-minute mark here. I'm going to wrap it up here. We did get some progress done today. We got some basics of this whole bio process going. And we'll see what else it's going to take here. But after this genetic stuff, we should be able to get our Varrox. And growing them, we should be able to produce our... Is it... some cocoons. And from those cocoons, we should be able to make... What is it now? I'm forgetting already. Alright, we need to make this stuff. Urea. Right, we're needing to get to methane, right? For formaldehyde. Formaldehyde, we need methane. So that's not gonna do. Which means we're gonna need the moondrop. Moondrop has a lot of recipes. The main one, we're gonna need this. We're just gonna need cDNA. Right, the retrovirus, that's what we need the Varrock cocoon for. That's right. Once we get that stuff shouldn't be too hard. Yep. Yeah, we've made all these things, the plasmids, Sogna, easy enough. Yeah, we're gonna need to finish genetics for that. Laboratory instruments, that's right. Rubber, just gonna take some stuff there. We'll be making organic solvent. We could do that recipe. Or we need coal processing too for that. So it'll need to be this recipe. Latex, carbon black, polybutane. Oh, that's not hard at all. Butylene. Not butane. Okay. Anywho, I hope you enjoyed watching, and we'll see you again next time.